So we're gonna have a look at the circulatory system in mammals. So I'm gonna I broke it up into sections. So we're gonna do it in different sections. We're gonna talk about the structure and function of the circulatory system that is outlined by the syllabus, right? And then we're gonna be looking at the cardiac cycle's electrical impulses, separating apart from speaking about diastole and systole of the heart. And then we're gonna be looking at blood pressure, right? And the effects of blood pressure, right? Or how to, whatever with blood pressure. And then we'll have a look at Bohr effect at the end, right? So let's start with the structure and function of the different parts of the circulatory system, right? Yeah, so let's start with that. So we're gonna start with the heart. No, it's very, very quite simple. Everybody knows the heart. Everybody should have, well, majority of persons would have done the heart lab, right? So we did a heart lab where we dissected a cow heart and draw the transverse section and the cross section and everything, the longitudinal and latitudinal sections of the heart. So some schools will have been able to do that heart lab, right? I thoroughly enjoyed the heart lab, right? Hopefully everyone did that, right? So this is a picture of one of the hearts, right? This is a much smaller though. Go ahead, Alan.
I'm just saying I looked from somebody's old lab and draw that. I've never seen that thing before.
You've never touched a heart, you never felt an aorta, you have never put your finger into the left ventricle.
Never.
What you
I looked at somebody's old lab and drew that. Okay?
mean?
I don't
Okay,
know what you're talking about.
okay. But let's go through it, right, the entire structure. So we know we have the chambers of the heart. This may be a snooze fest because everybody know it by now. Alright? So we're gonna have the atria and the ventricles. The atria sit on top of the ventricles. We have the right atria and the left, well, the right atrium and the left atrium, right, and the right ventricle and the left ventricle. Now the right atrium would receive the oxygenated blood from the venae cavae, right, and that's just pluralized version of the vena cava, whether it be the superior or the inferior vena cava, right, and the left atrium would receive oxygenated blood from the pulmonary veins. Lovely, right? Ventricles now, we have the right ventricle which pumps the oxygenated blood towards the lungs via the pulmonary trunk, right? And we're gonna have the left ventricle which pumps oxygenated blood, right, through the aorta into the systemic circuit, the coronary circuit, or, well, the systemic circuit or the coronary circuit, right? The pulmonary circuit would have been covered by the right ventricle, all right? And we know the circuits, right? We know the circuits. So the systemic circuit would have contained from the aorta, you have the carotid arch and the jugular vein. We have the subclavian arteries and veins. We have the carotid arch, well, not the carotid artery and veins, but the subclavian. We have the different types of veins, we have the hepatic artery and all these different type of arteries. Hopefully you guys spoke about it, right? I remember teaching C-sec that some years ago. All right. So we have different types of stuff. Right. The coronary circuit would have been with the coronary artery and feeding blood directly to the heart. Right. And then with the right ventricle would push it along the pulmonary circuit, which is between the lungs and the heart. Right. So the heart valves now we have the atrioventricular valves of the tricuspid valve, which is between the right atrium and the right ventricle, right here. And the bicuspid or the mitral valve between the left atrium and the left ventricle. Go ahead.
Sir, I have a question. Is the proper name for the mitral valve the bicuspid valve, or does Cape want us to say mitral?
So it's the bicuspid valve, right? Another name for it is a mitral. Mitral is usually used, but it is the bicuspid, right? Anatomically, it's a bicuspid valve. And then we have the semilunar valves, which are the pulmonary valves, right? And the aortic valves, so the pulmonary valves here and the aortic valves here, right? All right. We have, give me a second before I look at something. So I should have mentioned this before leaving the chambers of the heart. The left ventricle has a larger myocardium, right? Why is that? A larger section of the myocardium. Why is that? Why is the left ventricle larger and more
Because
muscular?
it's pumping blood towards the body. Therefore, it needs to pump with more pressure for it to reach the different parts of the body and organs.
Okay, lovely. While the right ventricle is smaller because it's only going to one organ, and what organ is that?
Lungs.
Lovely. Right, so the layers of the heart, we have the endocardium, the myocardium, and the epicardium. Right, so the endocardium is the inner layer of the chambers covering the valves, etc. Right, then the myocardium would be the thick muscular layer. Right, the myocardium, that would be larger on in the left ventricle. Right, so they're responsible for the contractile function of the heart, the contractions. Right, and the epicardium would be the outer layer of the heart. Right, it's also part of the pericardium which protects the heart. Right. We're gonna have the visceral pericardium and the peritoneal pericardium, right? So the pericardium here, right? So the pericardium is the surrounding of the heart, the double-walled sac, right? The pericardium of the fibrous pericardium and the serous pericardium, right? The fibrous pericardium is a tough outer layer that protects the heart and anchors it to surrounding structures. And then with serous pericardium, which houses the peritoneal layer and the visceral layer, right? So we have those two layers in there, which are part of the epicardium, right? And between the layers now we have pericardial fluid, which reduces friction during heartbeats. Everything is fine so far? Everything fine so far? Okay, lovely. Seeing the thumbs up. Alright, so now arteries, we know arteries are blood vessels that carry blood away from the heart, right? For the most part. But yeah, let me just say for the most part, because the umbilical artery crazy. But away from the fetal heart, right? But we talk about all of that in module 1. Not module 1, in unit 1, right? So the arteries themselves usually are the larger, more muscular, pulsative vessels in mammals, right? They usually have 3 layers. Usually the arteries, right, and the veins have 3 layers, right? They have the tunica intima, the tunica media, and the tunica externa, or what we call the tunica adventitia, right? For the purpose of this lecture, we're going to be calling it the tunica externa, all right? So here are the general structures here. So here in the tunica intima, we realize that there's a thin epithelium and some connective tissues. Here in the epithelium, however, right, we're going to have hematoreceptors. It is not in the diagram, but I'm most definitely going to make sure that it is mentioned, right? We have hematoreceptors, right? We have the tunica media, right? Here, which has involuntary muscle and elastic fibers, right? So muscle fibers, right? Muscle cells, right? Usually we're talking about smooth muscle, right? At CSEC we talk about 3 different types of muscles. Right here we're talking about smooth muscle, right? And then we have the tunica adventitia or tunica externa, where we have the external elastic membrane, and we have connective tissue within the tunica media and some parts of the tunica externa. We're going to have the baroreceptors, right? Primarily within the tunica media, we have baroreceptors. And with the tunica externa, we're going to have what we call the vasovassorum, right? Which provides nutrients to the arteries themselves. All right? Lovely. So the tunica intima, it has the epithelium, the subendothelial layer, and the internal, right, elastic lamina. So these things are important. So the epithelial layer is usually a single layer of epithelial cells, right? And they are directly in contact with blood flow. And so we have that there, right? With the subendothelial layer, is a thin layer of loose connective tissue just beneath the endothelium. And this layer contains scattered fibroblasts, right? And smooth muscle cells. We also have the internal elastic lamina, which contains some amount of elastic fibers for strength, allowing for the arteries to stretch and recoil, right? It appears as a distinct wavy line in histological sections and is particularly prominent in larger arteries, right? Then we have the end, and then we're looking at the role. The endothelium is there, right, to create a frictionless surface for blood flow, right? It minimizes any type of turbulence, right? But remember, it also contains chemoreceptors to measure the pH of the blood. right? The subendothelial layer now, the main purpose of there is just to add flexibility. And then the internal elastic lamina is generally there to stretch under pressure, right? So it stretch under pressure and it can return to its original shape, right? And also aids the maintenance of blood pressure and flow, right? So looking at the baroreceptors, chemoreceptors generally, right? Within the tunica media now, we have smooth muscle cells, elastic fibers, and the external elastic lamina, right? So here we're generally talking about the large muscle fibers that allow for the thickness of the arteries, right? And usually arteriodilation or contraction, right? So we're talking about contraction and those type of things, right? So looking at the interspread muscle fibers, right, that allows for the elasticity of the artery and also the strength of the artery walls, right? So we're looking at those type of stuff, right? The external elastic lamina, right, is usually just it separates, right, the tunica media from the tunica externa, right? So we're gonna have an elastic layer between the tunica media and the tunica externa, right? So looking at all those type of stuff. Within the smooth muscle cells, we have regulation receptors, bioreceptors that regulate and control blood pressure, right? And then the smooth muscle cells generally, right, are there for vasodilation, vasoconstriction. All right. Looking at those general things. Elastic fibers allow for the arteries to expand and contract with heartbeats, and the external elastic lamina, right, adds structural integrity and elasticity to the artery wall. Okay, you have the tunica externa, right? So it has connective tissue, a lot of connective tissue, and adipose tissue, right? So this layer consists primarily of connective tissues, and this is the layer in which we're gonna have the we're gonna talk about it, right? So we have that specifically, right? So we have a lot of elastic tissue, fibroblasts, collagen fibers, those type of stuff, right? And it has small blood vessels called the vasovassorum, which supply blood to the artery walls. It also has adipose tissues, right? So in some arteries, especially those located near fatty tissues, the tunica externa consists of adipose cells or fat cells, right? So the connective tissue there are important for the protection of the arteries, and the collagen adds to any type of strength, right? So speaking about tensile strength of the arteries, right, via the collagen fibers. The nerves now, right, so the presence of autonomic nerves in the tunica externa that allows the regulation of the smooth muscle and the tunica, well, in the tunica media rather. So it allows for the regulation of the contraction and dilation of the blood vessels, right? And also we have the vasovassorum, which supplies nutrients. So there are tiny vessels throughout the artery that provides nutrients to the cells of the artery walls, right? Particularly in larger arteries where the walls are really, really too thick for nutrients to diffuse from the lumen, all right? So looking at this, right, the tunica externa, the tunica media, which is the largest layer here, and then the tunica intima with a basement layer, and we have the lumen. Everything there is fine? I think the person's name who is Riri, I think your camera is on. Yeah, it is. Alright. So, alright, so that is generally the structure of the artery. Alright. So the baroreceptors, no, baroreceptors are located within the walls of our blood vessels, right? Particularly in the aortic arch and the carotid sinuses, right? So these are present to sense the changes in blood pressure, right? So when the arteries stretch, it activates baroreceptors. Conversely, when blood pressure decreases, less stretching occurs, leading to the decreased activation of baroreceptors, right? When the baroreceptors are triggered, it triggers reflex mechanisms to regulate blood pressure, right? So the baroreceptors detect high blood pressure. It sends signals to the cardiovascular center in the brain stem, right? Which in turn activates parasympathetic nervous systems, right? So the parasympathetic nervous system there, right, which their activity, right, inhibits the sympathetic nervous system, right, leading to vasodilation. So when the parasympathetic nervous system fires, right, it inhibits the sympathetic nervous system, right, from firing, right, and this leads to vasodilation and decreased heart rate. So it's resulting in decreased blood pressure. Conversely, when baroreceptors detect low blood pressure, we're gonna have increased activity from this sympathetic nervous system, which increases vasoconstriction and increases heart rate. So these things are quite important when it comes to blood pressure, as we will discuss. All right. The chemoreceptors now. The chemoreceptors are found within the carotid bodies and aortic bodies, right? And they are usually small clusters of cells located near the carotid arteries and in the aorta, respectively. Their function now is to figure out the levels of oxygen and carbon dioxide within the blood, as well as the pH or the acidity of the blood. So this is how we know if our blood buffers are not working. Alright? Now the response, now the chemoreceptors detect, when they detect a decreased level of oxygen levels, right? Hypoxia, it increases carbon dioxide levels, right? Well, or increase carbon dioxide levels, which is hypercapnia, right? So it can either, if there's a decreased number of oxygen within the blood, it's hypoxia, increased carbon dioxide, that's hypercapnia, or if we have decreased pH, we have acidosis, right? It detects all of these things, whether it's hypercapnia, hypoxia, or acidosis, right? This leads to an increased breathing rate, right, and depth, right, facilitating the exchange of gases in the lungs, right? So when there is a decreased amount of oxygen, which in turn will be an increased amount of carbon dioxide and acidosis in the blood, right, we're gonna actually have hyperventilation. The body is going to start to uncontrollably hyperventilate in order to take in as much air as possible to replenish everything, right? To replenish the oxygen back to its normal count, right? So we're gonna have that. So it generally happens when you have when you're not breathing, right? When you have suffocation, stuff like that, right? The blood acidity will increase, as we will speak about in the Bohr effect, right? And then the body will actually try to contract and bring all the air in order to replenish oxygen levels, right? And those things are baroreceptors and chemoreceptors are present within arteries. Lovely, right? For veins now, veins will be primarily speak to bringing blood back to the heart under lower pressures, and they usually carry deoxygenated blood apart from the pulmonary veins, which carry oxygenated blood to the heart, right? So they have also tunica intima, media, and externa, right? So here are the main differences now. So the difference between the vein and the artery specifically. So look at this, the tunica intima, it has an endothelium, subendothelial layer, right, and it also has valves. So the epithelium and subendothelial layer is the same as the ones that we talk about in the arteries, right? But the valves now, the valves are present to actually create, right, some amount of mechanism to stop the backflow of blood. So these valves are composed of epithelial cells and connective tissue, right? So they prevent the backflow of blood. Right? So we also have the endotheliums. We know that the endothelium there, right, is in direct contact with the blood, right, and it minimizes resistance of blood flow, prevents clot formation. The subendothelial layer provides structure to the endothelium, and the valves prevent the black flow of blood. Now it is important to note, veins are not as strong as arteries. They don't contain as much smooth muscle, right? So when we look at this, the media, right, they are much thinner than in the arteries, right? So we have that there, right? They still amount to some degree of vasoconstriction and vasodilation, right? But they need assistance, right? So the pressure within the venous system needs assistance, right? And that is from the smooth muscle within different parts of your body, right? So especially within the legs specifically, right? The legs are the furthest away from the heart, right? So the veins carrying blood from the when we're looking at the iliac veins, right? The iliac veins are coming from the legs, right? We're looking at those veins, they're carrying blood from the bottom, right, of the body, right, towards the heart, right? Now, since they don't transport blood under high pressure, and they don't have all those as thick as a wall or as thick tunica media as the arteries, their vasodilation and vasoconstriction will not help as much. So we're going to need our smooth muscle cells located within the lower half of our body to actually facilitate the movement of blood up towards the heart. So what happens now if we don't have enough movement, right? This is why we need movement and actually cardio exercises and stuff like that. If we don't have enough movement, right, within the lower part of our body, we're going to have blood pooling because of gravity. We're gonna have pooling within the veins in our legs, and we're gonna have what we call varicosis happening, or we call varicose veins, right? So usually when you see young persons who stand a lot or do a lot of traveling on foot, right, we're going or don't have a lot of movement, rather the ones that don't have a lot of movement, right, persons who don't have a lot of movement, usually persons who are a bit older, you tend to see large visible veins on their calves, right? And that is varicosis. That's varicose veins, right? Because there's not sufficient movement allowing for blood to be moving efficiently from the legs towards the heart. So varicose veins can occur. All right. So we have the tunica externa as well, which is not as well. It's generally around the same thickness as an arch. It's a little bit thinner, right? Really, the layer that changes is the tunica media. But with the tunica externa, however, it contains all the same things, right? Including a vasa vasorum, right? And adipose tissue. So it's generally the same thing, right? It's just that it may be a little bit smaller, right, than that of the arteries. Okay? So we have the difference there between the vein and the arteries. Notice that the tunica media is the one with the drastic difference, right, within size. And the vein will have a larger lumen, the artery will have a smaller lumen. Any questions or concerns thus far? Any issues thus far? Are we ready to move on? Alright. Just give me a couple seconds. Alright, so we've discussed the arteries and the veins, right? What's next? The capillaries, right? So they're the smallest blood vessels in the body. They're the primary sites of gaseous exchange within, or nutrient exchange as well within the tissues. Alright? So a typical structure of a capillary, right? Typical capillary is just have some endothelial cells, right? Usually one cell thick. So they have a single layer, right? So capillaries are composed of a single layer of flattened endothelial cells, right? In the that line the lumen of the capillary. They have tight junctions and intercellular clefts, right? These endothelial cells are connected by these junctions and these clefts, right? That allow for small molecules to pass through, right? And they have fenestrations. So we're going to look at the fenestrated capillaries, right? But generally within the capillaries, there are sites of gaseous and nutrient exchange, and they have some amount of barrier and regulation purposes, right? As they have selective permeability. Okay. Their general structure is a basement membrane, right? Thin extracellular matrix, right? The basement membrane is a thin layer of extracellular matrix, right? That supports the endothelial cells, and they're composed of collagen, laminin, which is another protein, right, which provides structural support and maintain the integrity of the capillary walls. All right, that's generally what it does. So we have the continuous capillaries, right, usually found within muscles, the skin, the lungs, and central nervous system, right, and they have few intracellular clefts, right, and they have an uninterrupted basement membrane. Right? They're the least permeable, allowing for selective exchange of small molecules and ions while restricting larger molecules. All right? Lovely. The fenestrated capillary now has more fenestrations, of course. They're found within tissues with high rates of exchange, such as the kidneys, intestines, and the endocrine glands. They allow for rapid exchange of water, small solutes, and certain large molecules. And then we have our sinusoidal capillaries, right? They have large gaps and discontinuous basement layers which create large openings, right? They're found within the liver, the bone marrow, the spleen, and the endocrine and some endocrine organs, right? They're highly permeable, allowing for the passage of large molecules, cells, and proteins, right? So those are general differences, right? We have the continuous capillaries, the fenestrated capillaries, and the sinusoidal capillaries. And that is the end of the structures of the blood vessels. right? What we're going to get into now is the structure of our blood cells, right? Any questions or concerns thereabouts?